Okay, thank you very much for your kind introduction. And also, I'm very happy to present our drug discovery project focused on the dengue, treating the dengue fever on behalf of the outstanding project team members. Dengue, widespread mosquito-borne viral disease, has become increasingly prevalent because of the climate change, urbanization, and also the increasing the international travelers. With almost half of the world population at risk, it is important to address the current limitation of the currently available dengue vaccine, which is age-specific and provide a limited protection against certain serotypes. And also that there is currently no dedicated antivirals available for the treatment of the dengue fever. Also, concerning the situation of the COVID-19, it is evident that both vaccines and antivirals play a crucial role in the reducing the managing and reducing the infection of the virus. Therefore, there is a currently urgent and unmet medical needs to develop effective antivirals for the dengue treatment of the dengue fever. Our approach in the direct antiviral is focusing on the reducing the peak viremia, leading to the faster clearance of the virus. By doing so, we aim to minimize the dengue disease symptoms and also the lowering the risk of the progressing the severe dengue syndromes. It is important to note that the dengue virus consists of the closely related but distinct 4 different serotypes, all of which cause the disease symptom. Also, the secondary infection with the different serotypes is known to increase the viral load and also increase the risk of the progressing to the severe dengue syndrome. Therefore, any developed antivirals should possess the pan-serotype activity against all 4 serotypes. Also, we aim to develop orally available antiviral drugs to ensure the patient compliance and also minimize the hospitalization requirements. Drug discovery process usually takes 6 to 8 years with the various stages from the laboratory research to the first in-patient clinical studies. My talk today as a medicinal chemist will focus on the hit to lead and the lead optimization steps, which are essential for the delivering of the clinical candidate. Before talking about a couple of case studies of these steps, let me briefly touch on how we begin this journey by finding a chemical starting point. We have taken the both target-based and phenotype-based screening to identify the suitable chemical starting point. In the target-based screening, our focus is on the viral enzymes, which are essential for the viral replication, such as the polymerase and the protease. To facilitate this, we developed a biochemical assay against these viral enzymes, which are suitable for the high throughput screening to enable us to rapidly evaluate a large number of the compound collection. Then from this biochemical screening, we aim to identify the chemical matters with the inhibitory activities against these viral enzyme activities. In addition, we employed cellular-based viral replication assays. These assays enable us to directly identify the compound with the broad with the broad range of the action, potentially including the inhibition of the non-enzymatic proteins, which cannot be identified from the target-based screening alone. Then, by combining the both target-based and phenotype-based screening approach, we can expand the exploration of the chemical spaces to enhance the chance of success to identify the promising chemical starting point, which may eventually lead to the drug candidate. Now, I'm going to talk about one case study, which is the target-based approach to identifying the dengue RNA-dependent RNA polymerase inhibitors. To understand the significance of the dengue RNA-dependent RNA polymerase, RDRP, this slide shows the dengue life cycle. So dengue virus entered the cell, then release the dengue genome plus RNA. Then plus RNA is used as a template to generate minus RNA by RDRP. Then plus RNA is replicated using the minus RNA as a template by the RDRP. Then resulting plus RNA is translated to the dengue viral proteins, which is assembled to release the new virus. As shown here, RDRP plays a crucial role for the synthesis of the RNA and also the viral replication. What RDRP makes attractive as a drug target is that RDRP has no mammalian counterpart and is the most conserved protein among the dengue viral proteins. Also, in the HIV and the HCV drug discovery field, targeting the viral polymerase is proven to be the validated target in the clinically. Viral polymerases fall into the two chemical classes, such as the nucleoside and the non-nucleoside inhibitors. Today, I'm going to talk about the target-based discovery of the non-nucleoside dengue RDRP inhibitors. We employed several target-based screening against the dengue RDRP. However, we faced a couple of challenges. Firstly, we see the protein is sensitive to the impurities such as the metal residues in the compound stock solutions, then leading to the many false positives after resynthesis of the primary screening hit. The other challenge is the previous biochemical screening resulted in the inhibitors with the poor physicochemical properties. For example, we found this sulfonamide compound as a primary screening hit with the IC50 of the 7 micromolar. Then consequent medicinal chemistry optimization to identify that this compound with improved biochemical potency. However, this modification increased the compound lipophilicity, which is unfavorable as a drug-like characters. And also, as I mentioned, we conducted several phenotypic screening. However, none of the screening hit from this screening has shown the inhibitory activity against the dengue RDRP. Then with this challenge, we decided to take fragment-based screening, which can provide small size starting point with the favorable physicochemical characters. Then we employed X-ray based fragment screening to identify the new chemical starting point for the RDRP. This slide summarizes how we proceed X-ray crystal, X-ray based fragment screening. So we screened a total of the 1,400 compounds in a pool of the 8 compounds. In total, the 176 pools were screened. And then compounds were soaked at 625 micromolar concentration. Then only one pool showed the difference of the density that indicated compound binding. Then further confirm the identified compounds were individually co-crystallized with the dengue RDRP for the further validation. Then this process identified that this biphenyl acetic acid as a screening hit with the IC50 of the 700 micromolar. Then this X-ray based fragment screening revealed the new pocket near to the active site, which is equivalent to the H1 HCB PAM1 site. More importantly, this newly identified pocket is conserved across all 4 serotypes. Now we started to the optimization of the screening hit to increase the potency and the binding affinity and the specificity to the dengue RDRP binding. So this is just summarize the optimization. So we started with the this fragment screening hit. Then introducing the carboxylic acid to enhance the binding affinity to this pocket. Then next, 6-membered phenyl ring was replaced with the 5-membered thiophene ring, which can potentially provide a suitable vector to fill the narrow inner cavity. Then we introduced several substituents to this position to fill the cavity. However, this modification didn't improve the potency. Then it was interesting to observe the water molecule exists deep inside the narrow pocket through the H-bond interactions. Then we try to interact this water molecule by introducing the nitrile group. However, this modification didn't increase the potency. Then, as an alternative approach, we try to displace this water molecule by introducing propargylic alcohol. This modification clearly improved the potency. Then we got the co-crystal structure of the compound 4 and the compound 6. This X-ray structure confirmed our hypothesis that propargylic alcohol displaces the water molecule and then have the H-bond interaction with the backbone of the priming group and also the H-bond interaction with the side chain of the glutamine 802. Importantly, these amino acids are conserved across the 4 serotypes. Therefore, this compound 6 shows the pan-serotypic activities for all 4 serotypes. Now our modification moved to this outer side to improve the cellular permeability to gain the cellular activity. So we removed one of the carboxylic acid, and then remaining carboxylic acid was replaced with the carboxylic acid bioisoster, such as the acyl sulfonamide, to identify the compound 14. This compound improved the biochemical activity. However, still inactive in the cellular-based assay even at the highest concentration. We got the X-ray crystal structure of the compound 4 with the dengue RDRP. This structure shows that this methyl group of the acyl sulfonamide doesn't have the interaction with the RDRP protein and exposed to the solvent side. This suggested us that we can modulate physical properties by changing this methyl group without affecting the potency. Then we introduced the different variety of the different substituent of the sulfonamide, and also that we changed the substituent of the benzene ring to lower the acidity to enhance the cellular permeability. After the several modifications, we identified the compound 17, which has demonstrated the single-digit micromolar against dengue 2 in the cellular-based assay. So next, to confirm that cellular-based activity is driven by the specific binding to the RDRP pocket we targeted. We generated the resistant replicon using the compound 17 and the compound 90 by testing the increasing the concentration. Then after sequence analysis, suggested that glutamine 802 changed to the aspartic acid, which has a one carbon shorter chain, resulting the losing the H bond interaction with the propargylic alcohol. In addition, we saw that leucine 512 was changed to the valine, which has the shorter side chain, then leading to the losing the hydrophobic interaction with the thiophene. Then we introduced this resistant replicon to the BHK cell to see to compare the cellular activity between the wild type versus the resistant replicon. As shown here, both compounds clearly show the EC50 shift in the testing the resistant replicon. This experiment clearly suggested that antiviral cellular activity is driven by the specific binding to the RDRP pocket we targeted. To our knowledge, this is the first chemical class to demonstrate a clear correlation of the RDRP binding and antiviral cellular activity. This is one story of the target-based approach to identify the dengue RDRP inhibitor. So now I will move on the phenotype-based approach to identify the clinical candidate. So as I mentioned, we conducted several phenotype screening to identify the suitable chemical starting point. However, we faced a couple of challenges. For example, many host factors are involved in the dengue life cycle. Therefore, this phenotypic screening captured the number of the compounds targeting the host factors. So addressing the safety concerns about the compounds targeting the host factors proved challenging in the optimization stage. Then second challenge was the some hit compound lacks the panserotype activity. For example, we identified the spiroindolin chemical compounds from the phenotype screening, which has shown the very potent activity against dengue 2 and dengue 3. However, this compound was inactive against dengue 1 and 4. And also we learned it was very challenging to change the phenotype character, such as from the serospecific character to the panserotypic character by medicinal chemistry optimization by phenotype-based medicinal chemistry optimization without knowing the structural information. Therefore, we took the most stringent filter to identify the viral target panserotype active compound. This is a flowchart, our most stringent filtration from the phenotypic screening. So 1.5 million Novartis compound collection was tested by the dengue 2 HH7CP assay at single concentration followed by the dose-dependent concentration to select 1600 screening hits, which were tested against dengue 1, 2, 3, and 4 serotypes, plus chikungunya assay as a counter screening. And also we tested this compound against the cytotox assay using a couple of different cell lines to ensure that compounds are not toxic in the host cells. Then after this screening, we selected 3 chemical classes which moved to the medicinal chemistry hit to lead optimization. Then we selected one chemical series for the further optimization. This slide shows the chemical structure of the selected lead compound, which has the cyanocyphene amide chemical series. This compound has shown the less than 1 micromolar potency against all 4 serotypes and also using the different cell line assays. And this compound has the sufficient cytotox window with the different multiple cell lines. This compound is stable under the liver microsomes and didn't show the inhibitory activity against the CYP and HAG enzymes. One issue was of this compound was the limited aqueous solubility we needed to address in the lead optimization stage. So this slide summarizes the story of the lead optimization process to the clinical candidate. So starting with this compound, then terminal sulfone was replaced with the sulfonamide. This modification improved the potency, however, still limited aqueous solubility. Next, to improve the aqueous solubility, we try to introduce the basic amine in the core to enhance the aqueous solubility. After the several trials, we identified the fluorobenzylamine here to maintain or improve the potency and also the increase the aqueous solubility. Then further optimization to balance out the potency, physical property, and the ADME profile resulted in the identification of the NITD688 as a clinical candidate, which has shown the single-digit micromolar, single-digit nanomolar against dengue 2 with improved aqueous solubility. Next, to confirm the mode of action of this chemical NITD688, we conducted viral resistant selection assay using the early lead series alongside the lead optimization, which eventually turns to the NITD688. So we tested that early lead compound at the higher concentration. Then we see the increase of the EC50. The certain virus shows the increase in the EC50, and then extracted this viral RNA was sequenced. Then we found that a mutation appeared in the NS4B region at the residue of the 195, 214, and 222. Then we see that these resistant mutation site is located between the transdomain 4 and 5 of the NS4B protein, or inside of the transmembrane domain 5. Then, importantly, these amino acid residue is conserved all 4 serotypes, but not conserved the other flavivirus, such as the Zika, West Nile, or Japanese encephalitis viruses. This translates to the NITD show the pan-serotypic activity against dengue, but not active for the other flaviviruses. Also, this profile also demonstrated our stringent screening flowchart led us to identify the pan-serotypic dengue-specific inhibitors. We also conducted an NMR study to confirm the binding to the NS4B using the recombinant wild type and double mutant NS4B proteins. So adding of the NIT688 to the wild type NS4B clearly shows the peak shift in the NMR study. On the other hand, NITD688 didn't show the peak shift in the double mutant NS4B. This study indicated that NITD binds directly to the wild type NS4B, but not double mutant NS4B. We also conducted in vivo efficacy study with the NITD688 using the dengue mouse model. We used two different dosing regimen. Firstly, to determine the minimum efficacious dose, compound was dosed immediately after the infection for the 3 days treatment using the BID dose orally. Then secondly, to simulate a more clinically relevant situation, compound was dosed after 2 days infection to see the efficacy. Then this slide shows the NITD demonstrated dose-dependent efficacy at 10, 30, and 100 milligram BID dose orally for the 3 days treatment. And also the NITD 688 still shows the more than 1 log reduction once a day treatment at 30 milligram per kilo oral dosing. And also in the delayed treatment protocol, this compound still shows a more than 1 log reduction at day 4 and day 5, 2 days post-infection. With this profile, so we selected the NITD 688 as a novel dengue antiviral as a clinical candidate, with demonstrating the pan-serotype activity with the 8 to 40 nanomolar across all 4 serotypes, and demonstrated efficacy in the mouse viremia model in acute and delayed treatment mode. And we confirmed this compound targeting the NS4B dengue protein. And also that this compound shows a favorable pharmacokinetic profile in rat and dog, and the compound was well tolerated in the animal safety studies. Therefore, that this compound progressed to the phase 1 clinical trial to confirm the safety profile in healthy volunteer. Now, NITD 688 is progressing to the phase 2 clinical trial to see the efficacy in the patient. Today, I'm talking about 2 case studies, which was conducted while our organization based in Singapore as the Novartis Institute for the Tropical Diseases. In 2017, our organization relocated to the Emeryville, California here as a part of the reorganization and integration to the Novartis Global Research Organization. In 2022, we were selected as one of the center of the NIH-funded pandemic preparedness program. Then over the last 2.5 years, we have been making the great progress and will make the best effort, hopefully to deliver the clinical candidate soon. Then we look forward to sharing the update and the progress in this forum sometime soon. Then now I'd like to close my presentation. I would like to thank all collaborators and team members who made a great contribution to the project I presented today. And finally, I'd like to thank you for your kind attention and engagement with my presentation. Thank you.